of different elements, given enough time, given chance, might just come together. But if it did, it would immediately be taken apart. And this is because today, our planet is just filled with the element oxygen. And oxygen breaks down organic compounds. Oxygen so-called oxidizes organic compounds. All that means is that oxygen pulls electrons out of organic compounds and turns them into inorganic compounds. So how could we even get the formation of any kind of organic chemistry if, as soon as anything even began to arise by chance, it was just immediately taken apart? Well, earlier in the 20th century, two scientists, Alexander Oparin, who was a Russian, and J.B.S. Haldane, a British biologist, independently suggested that the early Earth actually did not have much or any oxygen. Oxygen is all around us now in the atmosphere. But they suggested that when the planet was first formed, the first atmosphere that developed was entirely composed of just a few gases: hydrogen, methane, ammonia, and water vapor. This would be like the atmospheres of some of the outer planets that have been described, or the moons of the outer planets. And so Oparin and Haldane independently suggested that the problem of spontaneous generation of organic compounds probably wasn't as big a deal because the early Earth didn't have an oxidizing atmosphere. So to test this hypothesis, what Miller did was set out to reproduce the conditions presumed to exist on the early Earth before life had arisen, and to see if he could get the spontaneous production of complex organic compounds. Now, Miller's experiment was set up this way. He had two flasks connected by a series of glass tubes. He had a lower flask in which he put water, and he heated this water gently with a little flame, a Bunsen burner underneath it. That heat would cause the water to evaporate, creating water vapor, which would circulate up into a higher flask. Now, in the higher flask, in the upper flask, Miller also added a number of other gases. He created an atmosphere of the early Earth consisting of hydrogen, methane, ammonia, and water vapor, just as Haldane and Oparin had suggested. Now, Miller additionally exposed the gases in this upper chamber to a lot of energy by putting two electrodes in that chamber that would create electrical sparks. His idea here, he knew that you needed energy to create any kind of compound, certainly organic compounds. And so the idea here was that this sparking mimicked the lightning that would happen on the early Earth. There's a lot of volcanism on the early Earth. There was a lot of UV radiation. So presumably there was a lot of energy, and what Miller did was use these sparks to create kind of a lightning. Well, this is actually a pretty simple experiment, and you can almost do this in your own house. All of these materials are easily available. You could make the flasks, and you could replicate Miller's experimental results, which were spectacular. In only a couple of days, he found he could synthesize a whole range of different organic compounds, including some very complex ones like amino acids. Well, the scientific community immediately set out to replicate this. Many people replicated the experiment, and it quickly became clear that depending on the starting conditions, that is, depending on what recipe of gases you would put in that upper chamber and so forth, it was possible to spontaneously, without any pre-existing complex organic molecules, or what we might say abiotically, produce all of the amino acids that are normally found in living material, a whole range of complex carbohydrates and lipids, and most intriguingly of all, you can create nucleotides, which are the building block molecules that form the backbone of nucleic acids, DNA and RNA. The implication of Miller's experiment then, and those that followed, was that there appeared to be no trouble at all for complex organic compounds to rise spontaneously on the inorganic early Earth. And this is a first stepping stone to the origin of life from non-living matter. Now, on the other hand, we have to put this into perspective. As exciting as this result was, the organic compounds that Miller created were still relatively simple compared to the stuff that we're made out of. So what I want to do now is to ask, what else do we need, at a minimum, to get something from non-living material that we would call living? Well, for one thing, we have to take our synthesis of organic compounds even farther, beyond these organic building blocks, to get the very extremely complex molecules that living systems are really made of, things like proteins, DNA, and so forth. More importantly, we need three additional things to happen. Even if we get all of the stuff that we're made of, we have to have some way of reliably making more of it. In other words, we have to have a way of storing and retrieving information about the structure of these biologically important molecules, because we can't rely on random chemical processes to produce more of them. We, organisms, can't rely on that. Second, living things need to have these biological molecules somehow aggregated and organized on a higher level. I mean, the stuff that life is made of are these complex molecules, but they don't just get all jumbled together. Instead, they're organized at levels of scales and in particular packages. So we need to figure out how these packages could start to get put together. To do that, we have to understand how order is developed, how it can be maintained by an aggregate of molecules. And the third thing that living things need to develop is a more reliable way to gain access to energy and to use that energy. As I told you, the synthesis of these complex molecules depends on a lot of energy. Miller used electrical discharges in his experiment to get that energy. Living things can't depend on lightning or volcanoes or anything like that to power themselves. Now, we're going to pick up on the question of the origin of life in the next lecture, and specifically start talking about how we can get more complex organic molecules. But what I want to do for the remainder of this lecture is describe what we're going to do across the whole series of lectures to you in the context of these properties of life, these general properties of life that I've just described. Typically, a first university course in biology would take the following approach. It would start by talking about chemistry, basic chemistry background, building up to organic chemistry, and then it would move progressively to bigger and bigger things. After we talked about molecules, we might talk about cells. After we talked about cells, we'll talk about how cells come together to form tissues, organs, organisms. We'll look at how organisms form groups and populations, communities of species, and so forth, on to the really big stuff like ecosystems. We're going to talk explicitly about that hierarchy of biology in the third lecture of the course. And it's a fine way to organize a lot of details, but it makes it difficult to focus on the broader unifying principles of biology that I think are more useful for providing an appreciation of the subject. So in order to better draw out these principles, what I've done is to organize this course in a fundamentally different way, dividing it into three sections, each centered on one of the main conceptual themes I just listed. The first segment of the course is about information and evolution. Now, I've added evolution to the title of this section because, as we'll see, the concept of information in biology is inextricably linked to the way that information changes over time, which is what evolution is all about. We're going to, in this section of the course, begin by looking at information-bearing molecules, how they store information, how they take that information and move it to useful forms as they build more molecules. We're going to then see how that information can be transmitted to other organisms across generations through reproduction. And we're going to look at the consequence for the way that it's transmitted across generations, for how that information changes through time, which leads to evolution. And we're going to look at the consequence of evolution for how organisms have been transformed over the entire history of life and led to the diversity that we see around us today. Now, in the second segment of the course, which I've called Development and Homeostasis, we'll look at the issue of how the complex organization of living things comes into being, and how this organization is maintained in the face of what are really considerable external and internal challenges to holding it all together. We'll begin this section of the course again by going back down to the level of the smallest things, back to the level of molecules, and we're going to ask, how do we control the way that those molecules might build more molecules. That's going to lead us to the question of how we build an organism. How do we go from a single cell, say a fertilized egg, to a huge, multicellular, complicated organism like ourselves? We're not going to talk about the details of how we get from there to here, but we're going to talk about the principles which are required to allow us or any other organism to do so. That part of the course is also going to take us into an area that biologists would commonly call physiology. Once we've got a complex organism like ourselves with all of these different parts, those parts have to somehow talk to each other. They have to know how to work together. They have to send information between each other so that they can sort of get things working in the right way. They have to be able to respond to the outside. They have to be able to take on challenges like parasites that might want to come in and use us for their food source. And that brings us to the third segment of the course, which I've called energy and resources. This is where we're going to ask, where is all of the energy that is needed to make living systems work coming from? More importantly, not where it's coming from. I'll actually tell you where it's coming from. It's all coming from the sun, ultimately. But how do living systems capture that energy? How do they store it? How do they control its use? Again here, we're going to start at the level of the smallest things. We're going to start with molecules that are responsible for capturing energy from sunlight, molecules that are responsible for converting that energy into a storable form, molecules that are responsible for taking that energy and packaging it in particular ways that real organisms can use. We can't just light a fire to get energy if you're a living thing. You have to control the use of this energy very carefully, and we're going to look at how that's done on the molecular level. Now, the interesting thing about energy and other stuff we're made of, I mean, we're not just made of energy. We have to get a bunch of other things to provide the building blocks that we're made of. The interesting thing about energy and resources is that the way that they're used by organisms ultimately determines the highest levels of biological organization. It determines, for example, how many individuals of a particular kind of organism can live together in one place. That brings us to population biology. It determines how different kinds of organisms interact, an area that we call community ecology. Most organisms would like to eat other organisms. And that leads then to the way that biological systems are structured on the higher levels, above the level of the organism, in populations, communities, and ultimately, as we'll see, in ecosystems, where these organisms of all sorts of different types have to interact together and somehow interact with the available resources on the planet. And that distributes them in particular ways. So finally, at the very end of the course, what I'm going to try to do is to summarize all of this information and take a look, at the very end, at where life might be going. You just have to look around about us now and see that there are a lot of interesting challenges. This is what Wells was talking about when he said that biology is having such an impact. And he only could have imagined the impact that it's having now. We can take organisms and modify them and make them be what we want. We can create new organisms de novo. We can use those organisms to modify the way the rest of the planet is working. All of this is fine if we understand what we're doing. But sometimes we might get it wrong, or sometimes it might get a little out of control. Some of the things that we are doing ourselves as organisms, that we are doing ourselves as the dominant species on this planet, are changing the planet in ways that we're not fully understanding yet. And so I want to end by taking this view of where we've been and projecting it onto where we think life might be going into the future. And that's how the course will end.